Developing non-traditional antibiotics promises to be a novel strategy to combat multidrug-resistant organisms. But would they work? Are they feasible, really? Well, we're having a real expert today to talk about this. The objectives of this podcast is to discuss the notion of non-traditional antibiotics as potential therapeutics against multidrug-resistant organisms, increase our knowledge on the clinical efficacy of non-traditional antibiotics, and deliberate on regulatory pathways and approval of such compounds. Welcome to Editors in Conversations. This episode is brought to you by Antimicrobial Agents and Chemotherapy Journal, available at aac.asm.org. I am your host, Cesar Arias, Editor-in-Chief of Antimicrobial Agents and Chemotherapy, and this podcast is supported by the American Society for Microbiology, which publishes AAC. Don't forget to check our latest issue, always great papers, available at aac.asm.org. Outstanding papers on resistance, pharmacology, antimicrobial agents, epidemiology, and clinical therapeutics. Joining me today to discuss these important topics is, a legend in the field, Dr. John Rex. He is the Chief Medical Officer of F2G Limited and the Editor-in-Chief of AMR.solutions, who has been really thinking about this for a long time and writing really important knowledge on this. Welcome to the program, John.
Thanks, Cesar. It's fun to be here with you today.
Okay, so one of the topics that we want to discuss today is this concept of non-traditional antibiotics. This is a concept that has been put forward for people to try to develop molecules that are within that category. So let's start explaining to people, what do you consider a non-traditional antibiotic or antimicrobial molecule?
Non-traditional is a funny term. And actually, I don't find the term to be all that helpful, believe it or not. Even that's the subject of the podcast, and we will cover that. But the term itself is a little bit distracting. And so let me approach it a couple of different ways to think about ways to use this language. One way to think about something that's non-traditional would be something in terms of just structure. A typical small molecule, stick figure, something you can draw. A non-traditional might be a large molecule, a phage lysine. You might say, well, that's not traditional. It's not what we usually use. But in truth, when you're thinking about therapeutics, it's less interesting to know what the structure is than what it does. And so the other way to divide up the idea of traditional from non-traditional is to think about the purpose of the thing you're developing. So a traditional purpose would be treat an infection. A non-traditional purpose might be, for example, preventing the acquisition of resistance, or maybe preventing the spread of resistance, or keeping your microbiome safe. Those would be very non-traditional purposes for a drug. And so when I think about non-traditional, I really have to sort of pause for a second and say, which part of that am I interested in, and do I actually care where I am in terms of that? And while I'm on the theme of language, there are some other bits of language that people use sometimes, like alternatives to antibiotics. That tends to imply something that's not going to treat an infection. Sometimes that's used as sort of a way that is almost like a superset somehow of all the possible things you could do, like traditional medicines and other sorts of things. I tend to treat that as meaning the same thing as non-traditional, and then you have to ask the speaker, what do you really mean by that? The words potentiator and enhancer sometimes get thrown into the mix of the idea of non-traditionals. And that's another one where you really have to ask somebody, what do you really mean when you say a potentiator enhancer? So when you come back to sort of wrap it all up, you say non-traditional. My next question to you is going to be, well, what actually do you mean by that?
So Dr. Rex has been editor of AAC, and, thanks to him, we actually have a lot of his thoughts in the instructions for authors. So what would be the Rex best way to call these molecules?
Well, I think, non-traditional it's not so helpful. You might as well just call it what it is. And really, we want to talk about how do you use it? And so we actually spent some time thinking about this a couple of years ago. And there's a paper in Nature Communications from 2019 where we laid out an idea of 4 possible uses of a product. And this actually applies to both traditional and non-traditional, but I think it's most helpful with the non-traditional sorts of products. And so the acronym is STAR, or STAR. And the letters aren't exactly in the right order, but if you take the letters S for standalone, T for transform, and R for restore, here you've got ideas about things that are once you've made them, you still have a classic medicinal. So let's take standalone. Standalone just means it's a drug. It's a thing in a syringe, and off you go. And it's got the speed and the spectrum such that you could use it as the sole therapy, as monotherapy for an infection. Penicillin, for example, is an example of a standalone drug. And you come to me with a new thing and say, well, it's a non-traditional because it's a phage lysine. That's not traditional. I say, that's fine. But really, what you're going to tell me is you're going to try, can you use it as if it were a penicillin, as if it were something that would directly treat. The other 2 categories in S, T, and R, transform and restore. Transform, let me take restore first. Restore, beta-lactam, beta-lactamase inhibitors. You've got a beta-lactam is getting beat up by ESBLs, and you come in with a new beta-lactamase inhibitor that inhibits the ESBLs. You've actually created a small molecule by restoring its activity. You've put it back to where it was going to go, and you could, I suppose, imagine a non-traditional that did something like that. I suppose, but a beta-lactamase inhibitor, the way that works, maybe there's another antibiotic where you're going to protect it. You're going to bring it back to speed, bring it back up to activity by the thing you add to it. And the idea of transform is similar to that. What if you had something that would take vancomycin and make it into a gram-negative antibiotic somehow? Pick anything that currently works only on bacterium X, and basically that's all it's ever done up until now. You say, well, I am going to add something to it that causes it to penetrate, or whatever. But at the end of the day, in standalone, transform, and restore, what you've got is a thing that we all are familiar with, in the sense that you say, here's the dose. You can use it to treat patients with these bacteria and these syndromes, and you can use it to treat. It's a thing by itself. And at the end of the day, it is a standalone agent that you and I would be comfortable using because we can describe classically how to use it. So, and there are lots of non-traditionals that fit into that pattern. The phage, antisense technologies, but also traditional small molecules. The other corner of it, the 4th letter of STAR, is the one that I think attracts the most interest, and that's the letter that we call Augment. And here, an example of a non-traditional. This really would be non-traditional. Imagine it's called, how about an immunotherapy? Something that doesn't work on the bacterium itself, but works on you and causes your immune system to do something useful in the setting of the infection. Or an antitoxin, you could say. Well, I'm treating diphtheria. I need to have a diphtheria antitoxin, and I need to kill the Corynebacterium, right? So the idea of an augment agent would be something that comes in. It by itself is not a complete medicinal. It is, but it does make a classic antibiotic work better by doing something on the far side, if you will. The classic antibiotic's got to do its deal, but the augment agent comes in and cleans it up. Maybe it's an antitoxin is one of the easiest ones to talk about, but things that did something at the immune level, you could also envision. The STAR paradigm is helpful because it tells you something about what you have to do to develop the drug. For the consonants S, T, and R, you can actually do a classic development program. I can do a standard program with a new beta-lactamase inhibitor where I compare it to an existing agent and demonstrate to you that it works correctly in a standard non-inferiority design. And at the end of the day, you can get that approved. The issue with the augment agents is that by themselves, generally speaking, they're not a drug, and you wouldn't be willing to use it by itself. And so the design that you're forced into with augment is you're going to treat somebody with a good standard antibiotic at a good dose, And you've got to compare that with the same standard antibiotic at that same standard dose. And then you're going to add on your thing on top. And it's got to be better than the antibiotic by itself. It's got to be better clinically than the antibiotic by itself. And that's actually hard. Let's think about the math. For most of the infections that we treat, we do a clinical trial. Its typical success rates with typical endpoints are sort of around 80 to 90%. Let's just call it 85%, just to pick a number that's kind of a typical success rate in a typical trial. Community-acquired bacterial pneumonia, complicated intra-ab, you name it, that's kind of nosocomial pneumonia, that's where it's going to be. 85% response. That means that the good drug works 85% of the time. If I'm going to improve on that with an augmenting agent, I've got to do it in that remaining 15%. And that remaining 15% of people who didn't respond to the good agent correctly dosed for a susceptible infection, why didn't they respond? Well, some of them actually are never going to respond. You get people who are sick enough, nosocomial pneumonia, in the ICU. You can't save quite everybody. And so of that 15%, let's pretend that 5% there's nothing. We're real sorry, but you're not going to get there. So that really leaves you with a window of 10%. So in order to demonstrate that my augment agent takes a base agent and pumps it up, clinical improvement, measurable clinical improvement, I've got to do it within a window of 10%. And that is a big clinical trial. And also, you can say to yourself, what if I said, all right, it went from 85% response to 87.5% response? You might look at me, Cesar, and say, really? Yeah. Hey, do I really care? So that's the challenge with it. But that's why it's useful.
Let's go back to that point later, because I want to really dig into it, because you have put a lot of thought on this. But let's just... for the audience, for example, I just going back to your categories. Let's take a drug like one of the cleverest ideas that I've seen in the recently or in the last few years is that rivaxamase, for example, that giving you a beta-lactamase in order to break down antibiotics that get into the gut, like ceftriaxone, to try to protect your gut microbiome.
Right.
I mean, I thought that's a crazy idea, and that's very interesting that they have done it, right? So where would you put that drug in that star circle?
Well, you'd have to argue that it's an augment feature, because if what I'm doing is I'm taking an antibiotic to treat an infection somewhere in my body, right? And that I'm going to take this agent that is meant to destroy the beta-lactam before it gets down into my large intestine and thus protect my microbiome. What's the benefit I'm going to get from that? Well, my infection's over here in my arm. Okay, so I hope the drug still gets to my arm to treat my infection. The benefits in my gut, and maybe you're going to tell me, well, you're going to be less at risk for C. diff, or you're going to have less diarrhea or something like that. That's an augment feature, and you've got to actually, in a clinical trial, show me that I can feel that benefit. And in theory, less C. diff is something you and I could measure. We could agree that's a benefit. But there has to be something that we measure in the clinical trial that a human being can perceive. No one is interested in endpoints that revolve around a change in colony forming counts in the stool. You can't feel that. No one ever comes in and complains, oh my gosh, the E. coli in my gut has really been hurting a lot. No. You come in and say, I have diarrhea. And so you've got to know that there's something that you can pick up like that. Now, if you can, then great. But if all drugs that get developed, you've got to be able to say who benefits and how much is the benefit. And here it is. I've measured it. I did a clinical trial and I saw this benefit. And so you should pay for this because you're in the right category, and the benefit you'll get is X. You'll have less C. diff. So sure, you could do that.
Let's one of the studies, for example, for that drug was VRE. And as you know, I'm
It's
very
very,
familiar with that. And
yeah.
so VRE is a problem, particularly in cancer, immunocompromised patients. And VRE, what is called now not even colonization, but domination, could be a harbinger for bacteremia,
Yep.
right? So would it be feasible to design a trial to measure VRE domination as a clinical endpoint with a drug like that? Or would a company really consider that useful as a selling point, or will be just too weak? Regardless of the impact on mortality, because mortality is going to be influenced for so many factors in this population that is going to be very difficult to measure. So what would be your advice or thinking about studies in that realm?
You're not going to like my answer, which is to say that no one ever comes in and says, Cesar, the VRE in my gut is just really hurting today. They just don't. And so the idea of a colonization-based endpoint is simply a non-starter at the regulatory level. You cannot get a drug approved. You would not be able to get that approved at the FDA or the EMA level. And let me say specifically, though, I've said FDA and EMA, you couldn't get it approved, but which might make you think it's a regulatory problem. Surely the regulators should approve this agent. It's actually not a regulatory problem because the regulators are just the first ones who ask you the pointy question. If I now come to you and say, Cesar, I got this thing that you should give your patients, and it's only going to cost, $2,500 per patient per month, and it's going to prevent their VRE colonization, you're going to say to me, John, show me what's the benefit to the patient, and why should I pay for that? If all it does is knock down the VRE you can measure in the stool, but the VRE infection rate isn't influenced, who cares? So really what you'd have to do, and this is the hard thing, is you'd have to do a study in which you showed me that what you believe is true. When I take this thing and I keep the VRE from dominating, then you can measure the impact and reduced rates of VRE infection. Then, or some other symptom related to VRE, some symptom that I can perceive.
So how about, and I'm gonna try to dig because I did not like your answer, of course.
I know you did.
So what if I show you studies that indicate that patients colonized or dominated with VRE will have more bacteremia, bloodstream infections with VRE, and their outcomes are worse, regardless if it's cause effect, right? That number one. Second, that you could use a surrogate marker, which is eventually VRE bloodstream infection in this patient, and demonstrate that there is a difference when you do that. And using a similar model like for vaccines, where you measure antibodies. In vaccine studies, you measure antibodies, and people think that has clinical correlate. In this case, maybe domination, less bacteremia, I think for the clinical point of view, as a clinician, will be incredibly helpful. Would you agree with that?
So parts of it, yes. So because you said bacteremia. And so I could imagine a study in which you said, I'm going to show that taking my thing prevents VRE bacteremia. That's a measurable, that's a clinically relevant endpoint because you don't document VRE bacteremia unless you've got a symptom or something. I mean, that's not a random event. And so you mentioned the idea of vaccines and antibodies. Well, yes, we do talk about antibody titers as correlates of protection, but we always tie them back to clinical prevention. You look at the recent COVID studies. We knew before they started doing the studies that they could induce an antibody response, but that alone is not enough. You've actually got to show me that it knocks down the disease level. So all endpoints for all approved drugs have to be tied to something that you can feel. So the mantra is you got to be able to, you got to show how it influences the way the patient feels, functions, or survives. Feels, functions, or survives. And show me one of those things. I'm with you. VRE bacteremia is something you don't so much feel the bacteremia, but you feel the fever, and you feel your hypotension. So, those are things that are definitely part of what you can see with VRE bacteremia. So I would say prove it.
So how about, and thank you for that, how about these composite endpoints, I mean, we've talked about before about this desirability of outcome ranking door outcomes that actually we've now used a lot in these COVID-19 studies where there is an ordinal ranking scores about clinical improvement, not necessarily mortality.
Mhm.
So what do you think about those tools to measure clinically meaningful outcomes in things like are difficult to prove in the traditional way as you started the podcast?
Well, let me just say there's no trick back door here. At the end of the day, you have to measure a clinically relevant signal and show an effect. And for this kind of an agent, what you're looking for, it boils down to basically it's a superiority design. I treat you with X, I treat you with X plus my thing, and I have some outcome that is superior with thing added onto my base case standard of care therapy. And DOOR is a way of articulating kinds of superiority. And I think DOOR is very interesting in that it's saying that not all superiority is limited to not dying, which is kind of a nice thing to say. You can not die and not have renal failure, and that's better than not dying and having renal failure. That's an example of the DOOR level, with and without renal failure. I'd rather not have renal failure. So in principle, you could do that. You could say, I avoid VRE colonization. I avoid VRE colonization with bacteremia. Well, VRE colonization is not something you can feel, right? Dialysis you can feel in the sense that I'm putting you on a dialysis machine. Things have definitely gone wrong for you. VRE colonization is not the same because, at least until you tell me otherwise, I do not say if patients who are colonized with VRE are at risk for these syndromes, but at the moment that they don't have VRE bacteremia, do they feel any different than anybody else? I don't think so.
Yeah, well,
unless you
probably not. But we are not asking the patients if they are feeling colonization all the time. This is not a way that we evaluate patients. We just look at measurable things like bacteremia with that organism or with other, and we think it's clinically meaningful. And so Eric Paymer papers on this clearly show that this concept of domination of the gut microbiome and manipulation could be something to do.
Well, and let me just say that you raise an interesting thought here. We're moving these days in endpoints to being very interested in patient-reported outcomes. Because as doctors, we tend to think in a kind of a narrow box in terms of the things that the patient can feel and they care about. But there are, and DOOR is an example of talking about subtleties of that. But there may well be other things. I mean, I would say that if you had some insights about the kinds of symptoms or other consequences of having VRE colonization, if you can tie that to something that you can define and measure in a patient-reported outcome tool, that's a legitimate finding, because anytime you can demonstrate a superiority phenomenon, that's a powerful piece of data. Now the question is the quality. What's the meaning of the superiority? If you tell me, well, the rate of diarrhea related to VRE goes from, one episode a month to one episode every 3 months, you might say, not so much that I care about that. But if you find something stronger than that, there's no reason why you can't pursue that. The trick is you have to find something typically that's a pretty big signal to have it punch through the clinical noise. And that's the hard part.
Yeah, excellent points. I wanted to hear your thoughts about something that you mentioned, therapeutic strategies to prevent resistance.
Yeah,
right.
I love that one.
And you go back to your same point. Let's say we think, for example, combinations of antibiotics is a strategy that people use probably to be more therapeutically efficacious, but also preventing resistance. And we've done it with organisms, the animal models. And
we do it with TB all the time, right?
Yeah, exactly.
Yeah.
But that's Is that, in your logic, a clinically meaningful outcome?
Once again, I'm going to frustrate you. We actually, so the short answer is no. And we actually spent some time on this at an FDA workshop a while back, because in principle, it's absolutely a it's a benefit, it's a clinical benefit, but actually, who's the person who benefits? It's a community level benefit, right? I mean, in theory, preventing me from acquiring a resistant strain, I might have a benefit. We actually thought really hard about this, and the thought model was that there was a group that did a study of travelers from Sweden who went to India for a month or two and came back to Sweden. And you can measure that the returning travelers have picked up at a certain frequency gram-negative organisms that carry high-level resistance markers. They've gone, they've been at a place where those are more endemic, and they come back and they've got it. And so we actually modeled the idea of a therapy whose sole purpose was to prevent you from going to, when you were in the country where the high level resistance, you wouldn't acquire the resistant pathogen. It kind of doesn't matter how it works. Just sort of imagine a hypothetical pill you take once a day that somehow prevents you from becoming colonized. So you come back and now you're not colonized. Or if I want to do a study in which I demonstrate the clinical benefit of not being colonized, because it's also the case that colonization of those travelers wasn't durable. They didn't have it forever. They gradually cleared. So there's a short-term window during which I'm reducing my rate of colonization. Ultimately, it goes away. Is it worth it to me personally to take a therapy? And the therapy that prevents the colonization probably has at least one side effect. There's nothing that doesn't have some side effect, right? Do I want to do that or not? And we thought about, okay, what's the infection we could work with? And we actually realized, well, E. coli, urinary tract infections in women, you could model it and say, we're going to prevent urinary tract infections due to resistant gram-negatives in returning female travelers. It's a big study. The number needed to treat is larger than you would care that I can remember right now. It's a good size number. And so, and yet it's a cool idea. I'd like to not, I'd like to be able to do that. I'd like to be able to do that in a general case. But it is, you start hitting these problems of demonstrating the clinical benefit of a product. And remember, every therapy you give has an adverse effect of some sort, and so you don't do these things lightly, and particularly things that you might do for an extended period of time. The toxicology work to recover something you take forever is a kind of a big deal. So I wish there was something easier, because I'm like you. I liked the idea a lot. And actually, I was the one who made up the case. I said, I want to discuss this at the workshop. Can I make up a case? And we actually went back and forth on it and developed this little storyline. And we all went to the end and we go, oh no, it's just, I don't know. It's harder than we thought. Oh.
Excellent points, and thank you for pointing that out. I want to go back at the or just talk about the current modeling of non-inferiority to test an antibiotic. And I am going to give you probably the devil's advocate opinion of this, in which you now have to demonstrate that the new drug is at least equal to the existing drug in safety and efficacy, and that's it. So what would you like to use a new drug that is similar to an old one, and what would you pay for it? And that will potentially alter the logic of the superiority trial for non-traditional antibiotics, because basically you will have to demonstrate it's the same as the old drug. So what am I missing?
You ask a question that many people ask. It's bad bugs, no drugs. I want a new drug for the bad bugs. And you're coming to me saying that you studied it here's my fancy new beta-lactam, beta-lactamase inhibitor combination that covers every form of multidrug-resistant Pseudomonas, and you've compared it with a high-end carbapenem, and you've shown it's non-inferior. Why didn't you do a superiority study? Okay, so I'm going to tease you back a little bit. Let's talk COVID for a moment. If you were in the ICU with COVID and they were about to intubate you, and I came to you and I said, Cesar, we got a new drug for COVID, and we want to study it versus nothing. It'd be versus placebo. You might say to me, versus placebo? You mean I don't get steroids? I don't get somebody's monoclonal antibody? I don't get this other stuff, which kind of sort of works. What are you gonna? Are you really interested in that?
Of course not.
Of course not. Okay. So the thing about non-inferiority is that it allows us to study the new drug before the old drug wears out. So let's think about this for a minute. The way that I do a study right now is new drug versus old drug while the old drug still works. So you've got a bad infection of your foot with MRSA. I don't study my new drug versus methicillin. I study my new drug versus linezolid or vancomycin, right? And I've proven that the new drug works just as well as vancomycin or linezolid works, as long as vancomycin or linezolid work. If my new drug is well chosen and resistance it's not cross-resistant to the existing agents, then when the existing agents fail down the road, my new drug still works. I mean, the development of resistance to the other drugs, provided there's no cross resistance, and I'm going to choose wisely, the old drug still works. So the advantage of non-inferiority is that it lets us safely develop a new drug today for tomorrow's infection. And that's the key to non-inferiority. So you're right. If I've got a fancy new drug for high-end Pseudomonas, I don't use it on every Tom, Dick, and Harry. I use it only when I really need it. But in order to develop it, I have to develop it in a setting where I study Pseudomonas. And I have to find, and the people in the control arm want a therapy for Pseudomonas. I need to do my best to give them a drug that works for Pseudomonas. So the study that I can do and that you would sign up for is I come to you and say, well, you've got this bad Pseudomonas infection. Here's the old drug. It should work because your isolate is whatever it is. Here's the new drug. Looks like it works too. We need to develop the new drug. And you as a rational human being can say, yeah, I could do that. People do that all the time. In the future, when the old drug wears out, the new drug is still there. And that's actually the reason to develop it. So non-inferiority, even though it's bothersome, people don't like it. And yet the alternative, when I offer you the alternative, I'm going to do a superiority study. You go, oh, no. I don't want to be in that arm that's not getting an active therapy. Because these infections are lethal. And it's not like I'm treating a migraine. In a migraine, I can do a superiority study of a new drug for migraine. You know, what's the worst thing happens if I put you in a study of my new drug versus placebo for migraine? What's the worst thing that happens? You have a migraine for 12 hours longer, one time, okay? But the upside might be that maybe I'm developing a new drug that's super fast or whatever the catch is. But as a migraine sufferer, I'd be in your study once. It's going to hurt me for 12 hours if I get the placebo. Okay, I get it. That's it. But if I'm treating you for pneumonia and I don't give you an effective agent, we know that your mortality rate goes up the longer you don't get a good drug. And I cannot reverse mortality. That's not a reversible adverse event.
But if you don't have placebo, you wouldn't know what drug worked and didn't, as shown in COVID many times. And probably when we didn't do that, then we don't know if it works or not. So don't you think placebo is something that is absolutely crucial in those studies?
In theory, there is no difference between theory and practice, but in practice there is. And this is the challenge. So no one is willing, the physician's not willing, the patient's not willing to be in the arm that is the nothing arm. Unless there is truly honest to Pete nothing. In which case you say, for the very first drug, for the very first time for infection X, where you don't know whether the new drug is toxic, you don't know anything about it. That's one thing. But when there actually exists some combination of comparators or some combination of standard of care, people want the best available standard of care. And that's not just the patient, that's the doc, that's equipoise. I can't randomize you to a study where I know that what I'm giving you in the comparator arm is not going to work. I've got to put together the best thing I can and compare it with my new drug. And so the instant I have something that works even a little bit, the whole dynamic, the whole people dynamic changes. And we're human beings. We want to take care of each other. And so I'm saying to you that the key to a new drug, the reason we do non-inferiority studies for the antibacterials is we actually know that they're with no drug there you die. We know for community-acquired bacterial pneumonia due to the pneumococcus, we know that the mortality rate of that is 15 to 20% in young people. 20-year-old kid, pneumococcal pneumonia, 15 to 20% death rate without an antibiotic. We've got years of data on that. Somebody my age, it's a 2/3 death rate. So I don't have to ever again do a placebo-controlled study in pneumococcal pneumonia. I don't ever have to do that. We know that non-inferiority with modern designs detects inferior agents. It demonstrates the activity of new agents. There's no question about it. And the trick with new things is to gather that data as quickly as possible. And COVID is an example of how hard it is to develop placebo-controlled data, and why I'm going to say it's hard, and it's hard for a reason, because we really want to take care of people.
Fascinating conversation. So I want to sort of hear your final thoughts about innovation in this space, particularly in clinical studies. One of the things that we have seen in this pandemic is innovation in many ways.
Yep.
That recovery trial strategy is simple, thousands of patients that potentially help that. The investment that has been in vaccines, if you take the same model on antibiotics, would that get there? So what are your thoughts about that particular innovation in this space for this?
Well, so a couple of levels of the innovation game. One is just the level of innovation at the science of new therapies. And here I am really pretty excited about what I'm seeing in the early portfolios. There's been a lot of government money that's gone in through CARB-X and Novo Impact and JPI AMR. And we're seeing there's a large collection of really innovative small molecule and some non-traditional approaches that are out there bubbling around, trying to get into phase 1. If you look in the clinical pipelines right now, phase 1 and beyond, isn't so much innovation yet, and that's because it takes a while for the innovative preclinical stuff to get going. And we really started funding it seriously 4 or 5 years ago when CARB-X was created, 2016, I think. And so the truly novel molecules, the truly novel non-traditionals are coming. They're just not yet in the clinic. Most of the things will fail, but the few that make it, and there's some that I think I've got my fingers crossed. So that's innovation at the molecule level. And, if you will, COVID's an example of that as well. We were just delighted to discover that somebody had been working on the early stages of vaccines and the whole mRNA technology. Fantastic. And then there's innovation at the clinical trial level. And, it is, individual agents are still very much, a step-by-step kind of, it's a whole hand tool development program for any given drug. The idea of platform trials, the idea of, collaborative large-scale trials, we have debated that a lot. Hadn't happened yet, and there are reasons that sort of have made it, things that have made it difficult to do. But there's nothing about that is intrinsically undoable. I spent some time working with Wellcome Trust a couple of years ago, developing the description of a platform trial for nosocomial pneumonia studies, so that a new drug could just kind of get plugged in and get progressed through and you develop the data. And you can write it down on paper. It just costs money, and it takes people willing to support it. So, and then you mentioned DOOR earlier. That's another example of innovation. It's innovation on endpoints and the quality of things that we measure. So there is real innovation coming. It takes time for it to filter through. It takes time for us to learn how to use it. But I think we have seen a remarkable amount of innovation of filtering through, and I'm excited about actually what's coming in the antibacterial pipeline. 5-10 years from now, cross your fingers, we may actually have one or two really novel mechanism agents, if not registered, then in phase 3. I mean, my fingers are crossed.
Your final thoughts on the regulators. Innovation in regulation is a tricky word and difficult to move the needle, particularly when FDA, EMA has very clear guidelines on this. So how flexible do you think the regulators are going to be for those really wonderful innovation that might happen at the science level?
My experience is the regulators, they are as interested as we are in innovation. And they will, strong signal, clear new idea, they will absolutely work with you on it. There are a whole lot of ground rules. Medicinal has to be, the manufacturing, a whole lot of stuff you gotta know, because you gotta ultimately reduce it to a set of directions that Dr. Busy can follow reliably day in and day out. And so you gotta follow the rules of the road. But within that box, I have been impressed at how hard our colleagues at the agencies have been willing to work on thinking differently. And FDA has certainly had a whole series of workshops, as an example, where the question has been, what could we do that would actually move the needle and enable the progression of new agents? So, but the issue is the answers aren't easy. It's not like there's some little secret trapdoor that we've been ignoring. It's not. It ends up being a lot of little things that you have to adjust and tune. If there was a super easy way to make it quick and easy to develop a drug, we'd all do it. There's not some trick that we're overlooking.
With that thought, I'm going to finish the podcast. I want to thank you, John, for this wonderful conversation, enlightening, and the beautiful debates about this. I mean, it's really very useful information. I really appreciate it. This is Cesar Arias, Editor-in-Chief of AAC, signing off.